class. So this week we will learn on a new chapter, which is chapter 5 about prokaryotic and also eukaryotic cells, in which for today you will cover subtopic of microscopy, the importance of compartmental organizations, and also the nucleus. Now, what is organisms and also what is cells? Organisms are living things, okay? So all organisms are made up of cells. Meanwhile, cells is the smallest unit that can carry out all activities associated with life. So if the organism is made up from only a single cell, it is known as unicellular, such as protists, fungi, and also most of the prokaryotes. However, if the organisms is made up from multiple cells or million of cells, it's known as a multicellular. Usually plants and animals are multicellular organisms. So, we have to know also about the cell theory. Cell theory states that cells are the basic living units of organization and function in all organisms. And the cell theory also states that all cells come from other cells. So the evidence that all living cells have a common origin, which is provided by basic similarities in their structure and also the molecules which they are made. So they want to tell you guys that the cells each actually come from other cells that due to the similarities in their structure and the molecules that made up the cells. So, organizations of cells. The organization and small size of cells allow them to maintain the internal environment or maintaining the internal environment, also known as homeostasis. So to maintain the internal environment or homeostasis, the plasma membrane acts as a selective barrier between the cell contents and also the environment. Okay, we have a barrier here. Okay, and then most of the cells have internal structure. So the internal structure inside the cell is known as the organelles, in which they are specialized to carry out metabolic activities by their own or for the cells. So each cell has a genetic instructions coded in DNA, which is concentrated in a limited regions of the cells. Okay. If in prokaryotic cells, the DNA is inside the nucleus. Meanwhile, if prokaryotic cells, the DNA is located inside the nucleus. So, the cell size is limited and varied. So, everything that enter or leave the cell must pass through the plasma membrane. Okay, so the ratio of the surface area to volume is a critical factor limiting cell size. Okay, so if if the organisms is very small, so meaning to say the surface area is very small, right? Therefore, some variation in cell shape represent a strategy to increase the ratio of surface area to volume. So most of the protists or other bacteria are very small in size, and also the small size cause a smaller surface area. So usually their shape have been modified in which they do have a microvilli to increase the surface area of the organism. So, and then the cell size and shapes also are adapted based on their function. For an examples, we have immobile and white blood cells change shape as they move. Meanwhile, sperm cell, they do have a long and whip-like tail for a locomotion, for swimming. And the cells have long, thin extensions that enable them to transmit messages over great distances. And rectangular epithelial cells that you can find it on your skin and stack like a building block to form a sheet-like tissues such as our skin, and also the other linings of internal organ. So let's look on the microscopy. So microscope is the tool to study the cell structure. So the studying of the cell structure is also known as cytology. So the cell is actually content of many organelles, right? So the researcher use the technique of cell fractionation to isolate the organelles in order to study their function. So the early microscope was discovered by Robert Hooke, where it is the where he is the first one. He was the first one to describe the cells, and then Anthony discovered the bacteria, proteins, blood cell, and also sperm cell with small lenses that he made. And then in the late 19th century, the microscope was sufficiently developed for biologists to seriously study the cells. So scientists use microscope to visualize cells that are very small to see with the naked eye. So there are two types of typical or normal microscope, which are light microscope and also electron microscope. So let's look on the first type of microscope, which is light microscope. In light microscope, the visible light is passed through the specimen and then through the glass lenses. So the lenses will refract or bend the light so that the image is magnified. So this is how does the light microscope looks like. You usually can find it in a school laboratory and also undergrad laboratory. And this figure here shows important parts of light microscope. We have eyepiece here, normally can magnify the sample about 10 times, and then the prism used to reflect or refract the light source from here, but for light source, film, diaphragm, and also the lens. This is the mirror. Okay, to transmit the light here, the condenser, and here we're gonna put our sample on the stage and we hold it by using a stage clip, and then this is the lenses. So normally the lenses that objective lenses that you can find in your lab is with 10 times magnification, 4 times magnification, 40 times magnification, and also 100 times magnification. So if the eyepiece is 10 times magnification, and then you observe the specimen with objective lens of 4 times magnification, so the magnification is 4 times 10 is equal to 40 times magnification. And then, so there are three important parameters in microscopy that give the image a certain quality. So it depends on the magnification. As I mentioned earlier, magnification is the ratio of the image or object image size to its real size. Maksudnya berapa kali how to say, kita nampak kat spesimen tu kan, spesimen tu bigger than its real size, so we magnify, kita membesarkan lagi dia punya size tu. And the resolution is the measure of the clarity of the image or the minimum distance of two distinguishable point. Ini contohnya macam kalau awak gambar sesuatu and then you zoom the picture, right? If you zoom the picture and you can still see a distance or a space between two dots, meaning to say that camera that you use is a very high resolution. Okay, so boleh nampak clarity of the image. And then last important parameter is that contrast. Contrast is referred to the visible differences in parts of the samples. So kita boleh nampak the perbezaan in certain parts of the sample. Itu known as a contrast. So how does a microscope work? Based on magnification, resolution, and contrast. This one you can read it by yourself, and you can practice it during your experiment sessions. So next, microscopy can magnify effectively to about 1,000 times the size of the actual specimen, because the eyepiece is 10 times, and the higher magnifications of objective lens is 100 times. So 100 times 10 is equal to 1,000. Okay. So various techniques also used to enhance the contrast and enable the cell component to be stained or labeled. So most of subcellular structure, including organelles, are too small to be resolved by a light microscope. Therefore, a recent advance in light microscope used to improve the resolutions of the labeling cells. So such as a confocal microscopy and also the convolution microscopy will provide a sharper image of 3D tissues and also the cells. So let's look on the new techniques that been used in light microscopy. The first one is we have bright field technique. We have unstained and also stained specimen here. So an image is formed by transmitting light through a cell, and this technique is very suitable for observing the natural color of a specimen or the observations of the stained samples. Okay, and then the next technique in light microscopy is that phase contrast. So figure here show the detailed image of internal structure of living microorganisms, which is the animal cells, and the image in grayscale, and however the resolution is quite low compared to Nomarsky technique. In Nomarsky technique, the image appear nearly 3D image, okay, which is actually a 2D image, brightly color image, and the resolution is higher compared to phase contrast technique. And then next we have fluorescent technique, in which they use fluorescence to generate the image, as you can see here, and the visualizations of autofluorescence cell structure using the fluorescent stains. And this technique can greatly increase the resolutions of cells and also the cell structure. So if you shine the light on some molecules, you may see light of a different color, as you can see here, that being emitted from those molecules. So different molecules emit different colors. So this is known as a fluorescence. Okay. Next, we have confocal technique in light microscope. So confocal technique, sample or specimen usually prepare first by staining it with a fluorochrome. Okay, and exceptionally clear for 2D image. As you can see, the 2D image here is very clear. And then if you want to get the 3D image, it can be obtained by computer construction. Okay, this is examples of how does the confocal technique looks like. Okay, microscopy. So here is a microphage, okay, the white blood cells, and by using the XY slice, we can see a bacteria here in red in color, okay, inside the microphage cells. So next, we're gonna look on the second type of microscope, which is electron microscope. Electron microscope allow researchers to study fine details or the ultrastructure of cells. So usually these types of microscope will emit the electron flow, um, which is shorter wavelength compared to the visible light in that invisible light inside the light microscope, and this allow the electron microscope to have higher resolutions and also better magnification compared to light microscope. So some electron microscope have resolving power less than 1 nanometer. However, there is some advantages by using electron microscope, in which that electron microscope only can view the dead cells, not the living organism, because of the elaborate preparation required. Okay, and it also may introduce to structural artifacts. So there are two types of electron microscope. We have transmission electron microscope and scanning electron microscope. For transmission electron microscope, or known as TEM, the specimen is embedded in plastic and or thinly sectioned. Meanwhile, specimen in SEM is coated with gold or some other metals. So in TEM, the electron beam is focused through the specimen. Meanwhile, in SEM, they focus a beam of electron onto the surface of a specimen, so it will provide a 3D image. And usually, scientists use TEM to study the internal cellular structure. Meanwhile, SEM to study the surface. How does the surface of the specimen looks like? As you can see here, you can see in TEM, you can see the internal structure of the cells of the organism. Meanwhile, for scanning electron microscope, you can see a very clear 3D image showing how does the surface of the cells or the organism looks like. So this is how the electron microscope looks like. Okay. And this is the important parts that you have to know in electron microscope. Here we have high voltage of electric that used to short the electron gun here. The first condenser lens, and then we have condenser aperture here, second condenser lens, and then the second condenser aperture, specimen holder. This is where you're gonna put your specimen, and here is the objective lenses, the electron beam, and also the fluorescence screen, and also the camera. So this is how they focus the electron beam. For transmit electron microscope, they focus the electron beam through the specimen. Okay, so it will pass through the specimen to provide the image of the internal structure of the specimen. Meanwhile, for what we call it again? For SEM, okay, they focus on the electron beam onto the surface of the specimen. Okay, so that's why the image, the informations of the image will show the surface structure of the sample. So again, let's look on the two types of microscope that I mentioned earlier, light microscope and electron microscope. There are two types of electron microscope: transmission electron microscope, or TEM, and scanning electron microscope, SEM. So for light microscope, they use the light source that will pass through the specimen. Meanwhile, TEM, they focus on the electron beam to go pass through the specimen to provide the internal structure image. Meanwhile, for SEM, the focus of the electron beams onto the surface of the specimen, so it will provide the image of 3D image of the how does the surface of the specimen looks like. And then, as I mentioned earlier, scientists use the technique of cell fractionation to separate different types of organelle inside the cell. So, um, the cell homogeneity is subjected. The cells, okay, we have the cell that being homogenized will be subjected to centrifugal force by spinning the centrifuge. After you spin the centrifuge, you will get two fractions, which is known as a pellet and the supernatant. Pellet is referred to the solid parts or the solid portion, usually at the bottom of your centrifuge bottles, and a supernatant is referred to the liquid parts or the upper parts that you can see in your centrifuge bottle after you centrifuge the samples. So the supernatant is centrifuged again at a higher speed to obtain a pellet with the next heavier cell component. So pellet usually heavier compared to the supernatant. That's why the pellet will be at the bottom. And then the pellet can be resuspended and components further being can use to be purified by density gradient centrifugation. So if you want to separate the organelle inside the supernatant, you have to undergo differential centrifugation. But if you want to separate the organelle inside the pellets, so you can use the techniques of density gradient centrifugation. So let's look here. Here we have a tissue cell here, and then we homogenize it first, and then the homogeneity you put in the centrifuge bottle and you go the centrifuge process. Okay, so for the supernatant, okay, you can separate again the organelle that you can find in the supernatant, in which you pour the supernatant of the first tube into the next tube, you centrifuge again, you pour again, and then you pour again the supernatant into the next tube, you centrifuge again. So usually the supernatant that you will get, the pellet that you will get is based on their mass or their weight. Okay, so nuclei and cellular debris will become the first pellet because it is the most heaviest organelles inside the cells and etc. Next, so remember the cell have organelles. So each of the organelles, most of the organelles have their own internal membrane. Okay, so what does it mean by compartmental organization? So eukaryotic cells have larger surface area and volume compared to prokaryotic cell because eukaryotic cell is more complex compared to prokaryotic cells. So the average eukaryotic cell has 1,000 times the volume of the average prokaryotic cells. It's about 100 times of the surface area. And eukaryotic cells have more volume compared to its surface area because they do have internal membranes. So this internal membrane is referred to the membranes of the organelles. Okay? So plants and animal cells has most of the cells organelles. So what is the importance of the internal membranes or the cell compartmental organization? First, it serves as a dividing wall, and then it also have a unique lipid and protein composition depending on their specific function. And the internal membrane also participate in metabolic reactions since many enzymes are incorporated directly into the membrane. and it also provides a localized environmental condition necessary for specific metabolic process. For an example, um, the photosynthesis occur inside the chloroplast, so they have their own metabolic process that being enclosed by the membrane of the chloroplast, and also example the cellular respiration that occur in the mitochondria. And it also important for sequestration so that they may occur without the interference of other metabolic process from other organelles or from the cytoplasms of the cell. So this is how the animal cells look like. They have this flagella, nucleus, and which the nucleus have its own membrane, which is known as nuclear envelope. This is the plasma membrane of the cell, the ribosome. Here, rough endoplasmic reticulum, smooth endoplasmic reticulum do not have ribosome, and then we do have Golgi apparatus, mitochondria, microvilli, and peroxisomes, and three types of cytoskeleton: microfilaments, intermediate filaments, and microtubules, and also the cytosome. Centrosome here, it made up from two centrioles. And then this is how the plant cell look like. They do have the difference between plant cells and animal cells is that they do have chloroplast here, but plant cells do not have chloroplast. Okay, so I think that's all for today, class. Thank you. The most important organelle, which is nucleus. So nucleus is where the DNA located, and also it is contain the cell genetic instructions. So terms that usually related to nucleus are nuclear envelope, nuclear membrane, nucleolus, nuclear pole, nuclear lamina, and also chromatin. So let's look on how does it look like. So the inner core of nucleus is known as the nucleolus. So we have the chromatin here, and it wrapped by the nuclear envelope that consists of inner membrane and also outer membrane. And also we have a nucleopore here. So bear in mind that the nuclear envelope is connected with the endoplasmic reticulum, in which I will discuss about this one later. So nuclear envelope is the one that enclose the organelle, enclose the nucleus, therefore separating the content inside the nucleus from the cytoplasm, the liquid outside the nucleus. So nuclear membrane is a double membrane which is consists of inner and also outer membrane, and each membrane consists of a lipid bilayer. So this is how the surface of the nuclear envelope look like. As you can see here, nucleopore is very obvious. Okay, and this is a nuclear lamina. Okay, the one that located on the surface of the nuclear envelope. So the nucleopore function to regulate the entry and also the exit of molecules from the nucleus. And the nucleolus, which is the inner core of the nucleus, is located within the nucleus, and that is the location for the synthesis of ribosomal RNA. And the shape of the nucleolus is maintained by nuclear lamina, in which this nuclear lamina is composed of protein. So here. And how about the chromatin? Okay, this chromatin. So chromatin actually come from the DNA, as it contains DNA. As you can see here, the double helix of the DNA will wrap the protein histones Once it traps the protein histone is known as the nucleosome. And the stake of the nucleosome here is known as the chromatin fiber. And the chromatin fiber will coiling with other coiling of chromatin fiber, forming the chromatin, and then the chromatin will coil again within the chromosome, and this is how the chromosome looks like. Okay, so in the nucleus, DNA is organized into discrete units that call as a chromosome. We have X chromosome and Y chromosome, as you can see over here. Okay, so each chromosome is composed of a single DNA molecule that associated with proteins. The protein that I mentioned earlier, which is histone, and the DNA and histone are together. When it's called together, it is called as a chromatin. So when cell prepares to divide, chromatin will condense to form a discrete chromosome,